and 1. All right. So, somebody want to pray for us as we get started? Thanks, Davies.
Let's pray. Gracious Heavenly Father, we thank you so much for you gathered us here so we can learn about your creation, oh God. Pray that may you open our minds and our ears to listen and do as we learn about Father God. We thank you so much and we praise your holy name in the mighty name of Jesus Christ. We pray with all thanksgiving in our hearts. Amen.
Amen. All right. So our next set of questions that we need to deal with. What seems to be true of all eukaryotes? Okay, so we know eukaryotic cells have a nucleus. That's not true of prokaryotes. But what other things do eukaryotic cells always have, or at least usually have? What does the inside of a cell look like? What's unique about the eukaryotic nucleus? And how do cells transport materials around the cells? Now, last time we talked about how do cells ensure efficient transport inside and outside of the cell. And you know that that's by the plasma membrane and by transport proteins allowing things in. But let's talk about specifically within the cell. So what is true of eukaryotes? So in addition to the nucleus, which all eukaryotic cells have, all eukaryotic cells have at least some additional membrane-bound organelles. At least some additional membrane-bound organelles. Now, some of these organelles are found in some eukaryotic cells and not in others. And we'll talk about some of those as we encounter them. All right. So there are several organelles involved in normal cellular function. We've got the endoplasmic reticulum. What is its function? Anyone know? It does. Well, it's a site of attachment for ribosomes. It functions in...
Does it fold protein?
Yeah, functions in protein modification, especially protein folding. It also synthesizes lipids and some other biomolecules. The Golgi apparatus further modifies proteins, especially proteins that are lipoproteins, which have what attached to them? Lipids, or glycoproteins that have what attached to them? Glucose or other sugars, or nucleoproteins that have what attached to them? Nucleic acids. Those all get attached typically in the Golgi, or the Golgi body, Golgi apparatus, and then that also ships proteins to their final destination. Then we have mitochondria, known as the powerhouse of the cell. Now, the Golgi body and the mitochondria are absent from some eukaryotic forms. Neither of which are absent from any animals as far as we can tell, but there are some single-celled eukaryotic life forms that lack Golgi bodies, and there are some that lack mitochondria. Now, eukaryotic cells always have a plasma membrane with these specific protein components. No matter what the eukaryotic cell is, whether it's a liver cell or a neuron from your body, or it's a eukaryotic cell that came from yeast, a single-celled fungal eukaryote. There are always these three components. One, transport proteins. What are transport proteins going to do? Transport what? What's that? Materials into and out of the cell, right? Because things that are polar, things that are so polar that they're ionic, right, beyond polar, they can't move through the plasma membrane, except through transport proteins. Some of those are channels, some of those are pumps. All eukaryotic membranes also have receptors. What are receptors? Yeah, Davies.
They are stimuli that detects a change in their environment.
Yeah, they're stimulated or triggered by things in the environment. They bind to materials in the extracellular environment and start a response from the cell to that. Or sometimes they just bind to stick to it, and they don't trigger any response in the cell. And then the last one, the last component that you always find on eukaryotic cell membranes are labels. And these are basically a sign declaring what or who the cell is. And this is the way your immune system can tell self versus non-self. because it knows what the labels are on your cells. So any cell that it encounters that does not have those labels, it treats as foreign material. Which is interesting because you oftentimes have material entering your body that is not your own tissue. Every time you eat something, right? Or every time you drink water and you consume hundreds of microorganisms in the water. Okay? And so inside of your gut, you always have foreign material. Meaning what should not be very active in your gut? Your immune system. Okay, at least the part of your immune system that recognizes self versus non-self. Otherwise, it'd always be freaking out. All right. Now, some eukaryotic cells have cell walls. And remember, in prokaryotic cells, we said they have cell walls made of proteoglycan, this really sugary material. When eukaryotes have cell walls, they are not made of proteoglycan. Okay, fungal cell walls are made of chitin. Plant cell walls are made of what? Cellulose. Talked about that a few lectures ago. Do you ever wonder how we figure this stuff out? How many of you have seen a cell under a microscope? Pretty much all of you, if not all of you. A lot of these are extremely difficult to see, especially under light microscopy. And you're like, oh, it's so cool. We just use electron microscopy. What type of electron microscopy would allow you to see the inside of a cell? Transmission electron microscopy. But we already talked about some of the challenges there. What types of cells are you dealing with when you're doing transmission electron microscopy? Dead cells. So what about if you wanted to know what a living cell has in it? And so here's what they do to figure some of this stuff out. They do what's called cell fractionation. So they take whole cells. They're in a test tube. They're alive, living, just rocking life. Okay? And then what you do is you spin this cell at an incredibly high rate. And so you force it to experience a force 500 times the force of Earth's gravity. Right? If you've seen astronauts get on that thing that helps them to really experience really high G forces, and fighter pilots will do the same, right? So you get used to experiencing things that are like 6, 7, maybe 15 times the gravitational force of the Earth, 500 times the gravitational force of the Earth. And what that does is just rips the cells apart. And so whatever was going on in the cell, immediately at the time you ripped it apart, it's there. And then not only will that rip the cells apart, but it's also going to separate materials based on their size. When you spin something like that, what happens? Think about when have any of you ever driven in a car that had a balloon in it? Like a helium balloon. And it's floating in the car, right? Because it's lighter than air and it's just floating in the car. What happens when you turn left? Where does the balloon go? It goes to the right. Like this, it's so weird. And it goes fast. And you're like, well, I guess I mean, it's not physically attached to the car that's moving left, so it should go right. But why does that always happen? Why does it move so quickly? And it's because when you turn, you start to create some of these forces. The heavier materials move to the inside, the lighter materials move to the outside. And so the air is heavier than the balloon. It moves to the left, the direction the car is going. Balloon moves to the right. Okay? It also happens to you if you're not wearing a seatbelt, right? Because you're lighter than the car. Car moves to the inside, and then your inertia just keeps you flying to the outside. Anyways, so the heavier materials start to build down, and the speed at which you rotate it is going to start to separate first the biggest things, the nuclei. At 500 times the force of gravity, the cells rip apart, nuclei come to the bottom. And you're like, cool, there are nuclei in cells, and then a whole bunch of other stuff. Okay? Pull the nuclei out, or you dump what's called the supernatant, the fluid component, into another tube, and then you spin it at 20,000 times the force of Earth's gravity. Okay? And now you start to form a pellet of things that are a little bit smaller than the nucleus, but still bigger than like just general proteins operating in there, like mitochondria, chloroplasts, maybe the Golgi apparatus. And then you pull that liquid out and you spin it at 150,000 times the gravitational force of the Earth. And now you can get the smallest materials present in here. And you can see what was in a cell right at the moment that it was ripped apart. And you didn't have to kill the cell, wait around to coat the cell properly to look at it under microscopy, and then look at it. It's like immediately when that cell was ripped apart, this is what's there. David.
So even though you're spinning it and whatnot, the organelles are still alive?
I mean, you can spin it fast enough to rip the organelles apart before they'd have a chance to come here, but we're not doing that. Yeah, I mean, the organelles are still intact.
Oh.
Alive, the organelles can't be alive. Remember, the cell is the smallest unit that has all of the characteristics of life. So they're intact. I mean, you could destroy organelles, absolutely. And we'll do that in lab. Because sometimes you got to destroy those organelles to get access to what's inside.
Are we going to do self-fractionation?
We can, if you want to. We're not going to. I wasn't planning on it, but we absolutely can do that. Yeah, I mean, we have centrifuges to spin these things. We could get 20,000 Gs, no problem. 150,000 Gs, we'd have to go to a lab that has a room that can handle that. Because the force created by that centrifuge is immense. And you need a building that's reinforced to be able to handle that. So when you've got like an engineering lab in a building, the building has to be, because you're basically doing crazy things in engineering labs, right? You're trying to determine, could my building survive a magnitude 12 earthquake, right? So you're creating a magnitude 12 earthquake inside of this building. So it's dangerous. That's why you usually don't find really tall engineering buildings on college campuses, by the way. They're usually the really small. They're like surrounded by all these really big, immaculate buildings, and then you have this one-story, like, creepy-looking building, and it's because they invested a lot of money to reinforce that building. They're just going to keep using it. Sorry. All right. So then our next question, what does the inside of a cell look like? What does the inside of a cell look like? Now, different eukaryotic cells contain different combinations of organelles. We already mentioned that there are some eukaryotic life forms that lack mitochondria. There are some that lack Golgi apparati. That's kind of a fun way to answer this question. Well, it's going to vary. Right? It's like when somebody asks you the question of, like, how many calories do you consume in a day? You're like, varies.
Sunday yet.
Yeah, I mean, I have on occasion fasted. I don't know why, but I did. And so on that day, I consumed 0 calories. Then I've had 6,000 calorie meals. So I don't know, it just varies. The cytosol and the cytoskeleton, remember these, the cytosol is what? It's a fluid component. The cytoskeleton is the protein scaffolding. These two combine to form what? Cytoplasm. This is a lot more complex than what you find in prokaryotes and far more complex than what we typically think about. So when you try to picture the inside of a cell, what do you see? If I were to just ask you right now, describe what the inside of a cell looks like, what analogy might you use to describe it? Well, you'd want to use a factory, right? And you're like, okay, a water balloon, a factory, and you're like, okay, but this is kind of a weird factory because it's full of water, right? It's an underwater factory. And so what I don't want you to think of, I used to always picture the cytoplasm as just this big pool of water without a bunch going on, kind of like a pond or a lake. Right? And if you were going to move something across the cell, you just kind of hop in a rowboat and like row it across the lake, right? That's how I used to picture a cell. That's a horrible way to picture a cell because it really is. It's like an underwater factory or an underwater city. Incredibly complex. Materials going in all different directions. Different parts carrying out different tasks. Incredibly complex environment. I'm going to show you a couple of examples of eukaryotic cells. You've probably seen these before. In fact, you probably at one point had to build some kind of a model of them. When I was in the 7th grade, I had to build a model of an animal cell. And all of my classmates, they were making really lame models out of styrofoam. I mean, they might have been really good, accurate models, but I'm like, I'm going to build my animal cell out of edible materials. And so I built it out of Rice Krispie treats and all kinds of different types of candy. And then after I did my presentation, I cut my cell into a piece for every student in the class.
Wow.
You know why? Because I didn't care what the teacher thought about my cell. I was in junior high. I cared about what my peers thought about my cell, right? So here's an animal cell. A couple of things to notice right away. One, it has a nucleus. And you're like, cool, it should have a nucleus because it's a eukaryotic cell. Another thing to notice right away is we've got this really complex membrane-bound scaffolding off of the nucleus. That's the endoplasmic reticulum. Got our Golgi complex here, some more membrane. We've got mitochondria. Lots and lots of mitochondria. We've got a plasma membrane. Nothing outside of that. Now notice this plasma membrane is very, very simple, because what's not represented in this plasma membrane?
Protein.
None of our transporters, none of our receptors, none of our labels. Right? Here's a plant cell. Things you notice right away. It's got a nucleus, which is good. It absolutely should have a nucleus because it's a eukaryotic cell. Also has this complex membrane-bound scaffolding, the endoplasmic reticulum. It's got our Golgi complex. It's got mitochondria, but it has some things that we didn't find in animal cells. It's got chloroplast and a bunch of them. And what do plants do that animals don't do? They carry out photosynthesis. Here's where that takes place. And then it's got this massive structure in the middle, this central vacuole. You're like, wow, that's really weird. That's massive. And it's for a couple of things. One is these cells are physically attached to cell walls. And so if they start to shrink, and they'll pull away from this cell wall and it'll rip the membrane apart. Okay, so they have to work really hard to maintain water balance. Okay, so that's part of what this does. But they also, true or false, plants can photosynthesize 24 hours a day. False. Because what do they need to photosynthesize? Sunlight. And you're like, well, Dr. Engel, there are some places in the summertime where the sun never sets. That's true. There's some places where the sun almost never sets. But yeah, most of the day you're still not getting very good quality sunlight. So what do they have to do then? They're carrying out photosynthesis to make their own sugars. What are they going to do with those sugars? They're going to eat most of them, but they need to store them somewhere. And so you've got this massive storage repository as well. Yeah, Chris.
So if you had like plants in a greenhouse that were constantly
Given sunlight?
given sunlight, would the cell look a little different
That's a good question.
compared to like normal plants?
Yeah, it's a good question. Probably not so much, although it'd probably have more chloroplasts to make use of all the extra sunlight that it's getting. Its metabolic rate would be much faster than a typical plant cell, and so it would probably lower the lifespan of that plant individual. That's a good question. I don't know the answer to that. When you're doing a greenhouse and you're growing plants inside of there, you're really doing it to try to control the environment, right? You're doing it to grow something out of season, or you're doing something to grow something where you shouldn't ever be able to grow it anyways, regardless of the season. Like you're trying to grow plants in Dubai. You know, it's like things don't live in Dubai, but they do. Yeah, and so you've got to, man, if you don't want to ship plants there, I mean, you just got to figure out a way to grow them. Or if you want, to go skiing in Dubai, you just build an indoor ski slope, right? You just keep the temperature perpetually at 35 degrees Fahrenheit, right, just above the freezing point of water, and then you just keep pumping excess ice into there. It's just wonderful. All right. So before we carry out our next question, we're going to take a lecture break. And so what I want you to do is this. We did a lecture break. Maybe it was on Monday. I mean, maybe it was on Wednesday. I don't remember. These days, blend together for me because it's like all of my kids' birthdays, right? They all blend together, all of these great things in my life. That's actually not true, but I couldn't think of another analogy. So I remember every one of my kids' birthdays. You know what's interesting? I got to share this story with you. It really has nothing to do with this class. So if you're in the delivery room with somebody, they tell you not to look at that person's face because when they stop breathing while they're pushing, you'll stop breathing. And I'm the kind of person, if somebody tells me not to do something, I'm going to do that. Right? Because I'm like, you just watch me. I'm going to look right in her face and it's not going to affect me at all. And I was very, very wrong. So like my wife's been in labor for like 6 hours at this point, and now it's getting to a point where she stops breathing often, and every time I looked at her face, and sure enough, I stopped breathing. Like I wasn't even meaning to. I'm trying to keep myself breathing, but I stopped breathing, and then I actually almost passed out at the very end of the delivery, and my wife, in the middle of having our baby, just started making fun of me relentlessly, showing excellent ability to multitask. And she's like, why don't you have a seat since you're working so hard? Oh man, that was great. You should listen to that. You should not look at their face because sure enough, you'll stop breathing. And yeah, anyways, that was fun. All right, so, oh yeah, gosh, how did we get on that? So what I want you to do, I had you draw, without looking at anything, but just working with those around you, the plasma membrane, right? Do you remember doing that? I think it was it on Monday? Maybe it was on Wednesday. Wednesday, wow, that was just last class. What I want you to do now is I want you to draw the nuclear membrane. The membrane around the nucleus, not looking at anything, but just working with those around you, okay? I want you to draw what that membrane around the nucleus looks like. Take 90 seconds, starting now.
That's very, so good.
So that's 2, right?
And then it just says transport.
What was kind of...
I guess since you're having the flow of like
As you are describing, you know,
an hour of it, and then once like receptive one window of material, it should look like this. Assembled out of the where it has this little thing. Like that, like, is it gonna be like that again, or is it like?
Or we just wait a minute. So
Like, so like, you know what we're talking about? No, you know what we're talking about? No, do you wanna wait?
no, that's like she sat next to us like last semester.
Oh my gosh.
We'll wait. Just wait.
How do you know what's going on?
Just disconnect it. Just wait until
All right. What do we think? What's going to be there? What is forming the membrane? Let's start there. So what forms the plasma membrane? Phospholipids. What's going to form the nuclear membrane? Also phospholipids, right? If they make a great plasma membrane, don't you think they'll make a great nuclear membrane? They sure do. They make a wonderful nuclear membrane. Okay, so we're like, cool, we've got a lipid bilayer, just like we have on the outside. Which actually brings up some really cool questions, like what keeps them from combining together? It's another question for another time. Okay, what else is there?
pores.
What's that? They do. Pores? Yeah, because material needs to get out of the nucleus and material needs to get into the nucleus, just like the plasma membrane, right? Where the plasma membrane's got these transporter proteins, channels and pumps. The nuclear membrane has channels and pumps. It's beautiful, right? Because anything nonpolar is not going to move well, or anything, that is polar is not going to move well across that nuclear membrane. But we know we need to get stuff out. Okay, what else is there? Yeah. There aren't ribosomes. There are ribosomes in the attached to the membrane of the endoplasmic reticulum. But remember, we have 3 different protein components of the plasma membrane: transport proteins, receptors, and labels. What do you think we have in the nuclear membrane?
Label.
Transport membranes or transport proteins, receptors, and labels. And what's interesting is you have some very, very unique receptors on the nuclear membrane. Some very, very unique receptors. You also have some very, very unique labels so that it keeps the nuclear membrane from fusing with the plasma membrane. Very unique labels. All right. Bless you. So what are some of these things? Well, the eukaryotic nucleus, it facilitates separating the genetic material. So we take the genetic material and we can package it away from the rest of the cell. Then this is reinforced by structural proteins. So in addition to the transport proteins, receptor, labels, we also have some structural proteins called lamins in animals. There's lamin A, lamin B, and lamin C. Yeah. And that's nice because sometimes it'll skip, right? Well, there'll be like lamin A, lamin B, and then lamin D, and you're like, what happened there? What happened to C? And oftentimes it's because they name something a lamin, and then they realize that's not a lamin at all. So, but they are already taken up lamin C, but that's not the case here. Then we've got these pores called nuclear pore complexes. These allow materials to leave, like RNA. Right? We talked about the analogy of the factory and the nucleic acids. What type of workers are they? They're the white collar workers, but then you've got DNA that's like the white collar worker that never leaves the office. You don't know how they get in there. Right? There must be some secret entrance to that factory. They don't want to rub shoulders with anyone. The RNA, they're the white collar workers, but they rub shoulders with the proteins, right? The people that work in the factory. And so the RNA, they've got to leave the nucleus. Then we need materials to be able to enter, bless you, the nucleus. Proteins, other types of RNA. Now here was something really cool. This was like probably one of the coolest things that we've really figured out a lot about between when I learned cell biology and now. I think this might be one of the coolest things. There's proteins that need to operate within the nucleus because proteins are translated, and we'll see this in a few weeks, but you might remember this from seeing this before. Proteins are translated inside of the cytosol, okay, on ribosomes, or they may be translated on the rough endoplasmic reticulum. But anyways, if they need to get to the nucleus, at one side of that protein, they have what's called the nuclear localization signal. And it's basically a label on the protein that says, I belong in the nucleus. And then there are other proteins that recognize that signal, grab that protein, and transport it to the nucleus. So that it can end up being exactly where it needs to be. And you find this for proteins that operate in the mitochondria, where they'll have a mitochondrion localization signal that proteins will recognize, grab, and then take them to the mitochondria. That's cool, right? That's good stuff. That is good stuff. All right, one last thing about the nucleus. Within the nucleus, you often find a darker, really dense structure called the nucleolus. And this is the site of where we are transcribing ribosomal RNA. And we'll talk more about ribosomal RNA in a few weeks, but just suffice it to say, you make a lot of ribosomal RNA. And you're constantly making it to replace ribosomes that are damaged for some reason or in some way. All right. So here's a a cartoon of the nuclear envelope. Here we've got the nuclear envelope with these pore complexes. Notice in a lot of places we actually have a double membrane around the nucleus, not just a single lipid bilayer, but a second lipid bilayer. Here you can see the nucleolus, that site of what? What's going on there? Ribosomal RNA production. Yeah.
So is the chromatin like the DNA that needs to be used, does it get pulled through the nucleolus so it can be copied?
Yeah, so chromatin is a combination of the DNA and proteins. And so you actually have to synthesize, your cells have to synthesize the proteins involved in that chromatin. The only way to make a protein is to write the section of the DNA that codes for that into RNA. The RNA can leave, get translated into that protein, and that protein needs to be recruited back into the nucleus. Just like in a factory, because that white-collar worker sitting inside that room, they're not going to do anything, right? They just got to direct the flow of information and direct the flow of work. And so here it shows you basically a zoomed-in portion of this here. We've got this phospholipid bilayer, same way we did in our plasma membrane. Okay. So here is the research that was done to show the presence of this nuclear localization signal. So he took a protein that was regularly recruited to the nucleus, and then they started removing portions of that protein. So here the amino acid at position 128 was mutated to change it into a different amino acid. And look at that. Now the protein is not recruited properly to the nucleus. Deleting any of the amino acids from 1 to 126, that protein still made it into the nucleus. Now it wasn't functional because you started cutting away parts of that machine, but it still had the part of the machine with that signal to tell other proteins to recruit it there. But if you deleted any amino acids here after that localization signal, now that protein again could not be properly recruited to the nucleus. So there was a piece of that protein code that when that protein was folded correctly operated as a tag, a label for another protein to realize it needed to be in the nucleus, grab it, ship it to the nucleus. All right? Make sense? Any questions? Okay. So next question. How do cells transport materials around the cell? So there are two main ways cells can do this. What is within the cytoplasm that would work as a wonderful structure to move materials? The cytoskeleton, right? So cytoskeleton, we know we've got these protein components that are providing the scaffolding of the cell. That's going to be a wonderful way to move materials around the cell. But we have another wonderful way in which we can do this as well. And it's using what's called the endomembrane system. And so cells use this system to transport proteins and other materials all around the cell. What does that root endo mean? Inside. What does membrane mean? Membrane, right? We've got a plasma membrane, we've got nuclear membrane, we have other membrane-bound organelles. So they use this system of membrane-bound organelles to transport materials around the cell. Now this can also be used to bring materials into the cell. This process, the process of phagocytosis, when a cell actually wraps something with its membrane and then pinches that, what we would call this like a vesicle now, off. Could also be used to send materials completely out of the cell. We call that exocytosis. What does this root mean? Exo? Outside. It's the opposite of endo. What does cyto mean? Cell. Outside cell. You're like, that's so wonderful. The name means something. And so the system is composed of basically a series of membranes. Some of these are physically attached together. Some of these are only attached together by some cytoskeletal element connecting the two. Again, series of membranes either physically attached together or connected by some cytoskeletal protein that connects the two. These are all of the components of the endomembrane system. It starts with the nuclear envelope. The endoplasmic reticulum is part of that endomembrane system. The Golgi apparatus. Now these two are physically attached together. Endoplasmic reticulum is physically attached to the nuclear envelope. Golgi apparatus is separate, but they're connected by what? They're separate but connected by cytoskeletal elements. Okay, lysosomes, these are vesicles that digest materials. Vesicles are just floating blobs of membrane with something inside them. And then the plasma membrane. And this works as basically a one-way road. If something starts in the endoplasmic reticulum, it'll move to the Golgi body, then we'll move into a vesicle. Well, actually, it'll go from the endoplasmic reticulum into a vesicle, into the Golgi body, and then basically somewhere else. Like if it needs to be sent out the cell, ultimately to the plasma membrane. Or something can move inward. But things don't tend to move and then back up and then move and then back up. So the endoplasmic reticulum comes in two forms. The smooth form, not covered in ribosomes, and the rough form, covered in ribosomes. So if ribosomes are working on producing protein, what is the rough endoplasmic reticulum going to be involved doing? Yeah, modifying proteins. If the smooth endoplasmic reticulum is not covered in ribosomes that are working on making proteins, the smooth endoplasmic reticulum, what is it not going to do? It's not going to modify proteins. And instead, the smooth endoplasmic reticulum produces lipids, like phospholipids for membranes, and it also produces enzymes for breaking down different materials. So liver cells work really, really hard to detoxify materials that come from what you eat or drink. Okay? Basically, everything that gets absorbed by your intestines has to be shuttled through the liver before it can go into the rest of your circulatory system. Okay? So liver cells work really hard to detoxify these materials. So which structure do you think is really, really big in liver cells? The smooth endoplasmic reticulum. Because remember, part of what it does is to make enzymes that break down foreign materials. Okay. Proteins modified in the lumen or the inside of the rough endoplasmic reticulum, especially proteins that are going to be present in the plasma membrane. What types of proteins are present in the plasma membrane? Somebody remind me. I've forgotten from 8 minutes ago. What kinds of proteins? They come in 3 varieties that you find in the plasma membrane. Transport proteins, receptors, labels. Good. Lipids are made, toxins destroyed in the smooth endoplasmic reticulum. I already told you that. Sorry. I ruined the surprise. You may have been surprised when I told you, though, so then you got the surprise, just not at the right time. Thank you, Peyton. Now, proteins further modified, packaged in the lumen of the Golgi complex. Now, a lot of this modification is, again, attaching other biomolecule types to these proteins, forming lipoproteins, forming glycoproteins, or forming what? Nucleoproteins, which are a combination of nucleic acids and protein. Vesicles function to carry materials between the structures of the endoplasmic reticulum, unless, of course, the structures are physically attached together. Okay. So we're going to take another lecture break here. Give you a moment to write down these pieces of information. What I want you to do during this lecture break is this. I want you to work with those around you. Take about 90 seconds. And I want you to come up with the order of structures a protein interacts with on its way to its permanent home in the plasma membrane. Okay? So we've got one of these three protein types. Let's say it's a label. It's a label designating your cell as your cell. Right? So it's a label in my cell designating that this is Dr. Ingalls' cell. I doubt the label says that, but who knows? So what I want you to do is take about 90 seconds, work with those around you, and I want you to come up with the order of structures that protein interacts with or is found inside on its way to its permanent home in the plasma membrane. Okay? Take 90 seconds starting now.
Nuclear, yeah, so it gets first to no, so it goes to the ER, doesn't it go to the ribosome, so that the ribosome can copy it, and then it goes, because the NRA isn't used, it's used just to form the protein, it's like a blueprint, so it needs to go to the ribosome so it can actually make the signal. Yeah, I'll go to the ribosome and then to ER so it can get folded,
Also
right?
further modified proteins as well. Or well, the endoplasmic does.
Okay. Oh, that's
Further
right,
modified
yeah.
the
Oh, you know what?
and then I guess
I think, because lysosomes, I think they flip part of their body over like bad things that come into the cell, but I think they can also do that for transporting stuff, so maybe that's why they go to the lysosome,
All right.
so it can go to vesicles.
So we've mentioned this already a couple of times today, but what structure is involved in the process of forming proteins? Well, the nucleus, because it contains the information, but you really don't get a protein until you get the RNA out of the nucleus and to get it translated into protein. And what does that? The ribosome. Okay, so the very first structure that protein is going to interact with is going to be what? A ribosome. And now that's the first time you actually have a polypeptide. You have amino acids strung together as a polypeptide. Why do we call it a polypeptide? What does poly mean? Many. And what are peptides? Peptide bonds, the bonds that form between amino acids. That's good. Critical thinking at its finest, right? What's the next structure? So it starts with the ribosome. The rough endoplasmic reticulum. Okay? And typically, it's actually going to be translated on the surface of the rough endoplasmic reticulum. And as it's being translated and formed, it's being shuttled into the endoplasmic reticulum. It's kind of cool. We'll see this more in a little bit. Not today, but a little bit. I don't know how long. Then where does it go? So in the endoplasmic reticulum, it gets folded correctly. So now it's actually a functional protein, but now it's not at its final destination. It's only at its final destination if it functions in the endoplasmic reticulum, but it doesn't. I told you, where's the home of this protein? Plasma membrane. So where does it go from the rough ER? No, it doesn't go to the smooth ER. It goes to the Golgi, but it has to go to something in between. A vesicle. Okay? So it's got to go from the rough endoplasmic reticulum into a vesicle. Now the beauty of this is the vesicle is just a ring of membrane surrounding materials. Where do you think the material for that membrane came from? It came from the wall of the rough endoplasmic reticulum, which is made of membrane. And so you basically just pump that protein to one end of the rough endoplasmic reticulum, and as it pushes the membrane out, it rips it off of the rest, and that membrane wraps around to form this vesicle. It would be akin to, let's say, let's just say in theory you liked to go to Dodger games because you really enjoyed watching the team make it all the way to the World Series year after year after year, only to not show up, right? So you really enjoyed that. So it'd be akin to, you're here in Santa Clarita in the rough endoplasmic reticulum. You want to be inside Dodger Stadium, which is the Golgi body. It would be like a piece of the city of Santa Clarita, like this wall around it, wrapped around you, shuttled you down to Dodger Stadium, fused with the walls of Dodger Stadium, and released you inside. So that then you could watch the Dodgers make it all the way to the World Series, only to end up short. Now, I'm old enough that, if any of you are Dodger fans, you can feel this with me, but I'm old enough that I was actually alive the last time the Dodgers won the World Series. I was 1 year old. Yeah. But we have a picture of me with my dad watching the World Series on television, rocking a Dodger onesie. So I feel like it counts, even though I'm not old enough to actually remember it. Okay, so now you're in a vesicle. The vesicle will transport this protein over to the Golgi apparatus. Then the Golgi apparatus, and maybe further modified if necessary. Some of our proteins, especially since this is a label, oftentimes that protein's going to be more than just protein. It's going to be some kind of a complex like lipoprotein or glycoprotein. It's got to have some sugar or some lipid added onto it. Okay, now it's in the Golgi apparatus. Where does it go from there? Another vesicle. Because the Golgi apparatus is not physically attached to the plasma membrane. So it's got to go to another vesicle that will transport it from the Golgi apparatus to the plasma membrane. And the beauty is, the vesicles, they peel off the Golgi apparatus or the endoplasmic reticulum, then they go and fuse with the next structure in it. So this protein, if it's going to operate in the plasma membrane, it's got to be inside of the membrane of the vesicle, right? So it's inside of the membrane of the vesicle, so when that vesicle fuses with the plasma membrane, the protein's in place now. Yeah. And you're like, this is so cool. These vesicles are fusing with the plasma membrane, and that's so awesome. But then are you thinking, why doesn't the plasma membrane then just keep getting bigger and bigger and bigger until it becomes like this big old floppy pair of jeans? Like somebody that loses 300 pounds, but they keep rocking the same pair of jeans because they don't want to buy any new jeans, right? And they've got them like tied around in a knot on both sides, some hair ties. You seen that look? It's a good look, right? Why doesn't that happen? Because that doesn't happen. The plasma membrane never becomes an oversized pair of jeans. If that's the only thing you take home from today, take that home. Just be like, hey, you know what I learned today in class? The plasma membrane never becomes an oversized pair of jeans. It's wonderful. Tara. Yeah, because at the same time, phagocytosis is happening. That the cell's wrapping portions of its plasma membrane around material it wants to eat and pinching vesicles off inside of the cell that then go to the Golgi apparatus, empty out. Actually, they may fuse with the lysosome to get digested, then go to the Golgi apparatus to start shipping those materials all around the cell to where they belong. And so here is an image from your text. Is this from your text? No, this can't be from your text. This is from another text. This is a good one, but you have these slides. They're on Canvas, so even though this isn't in your textbook, you have access to it, because this is a good one. And it shows you the endomembrane system, and it shows you the order of materials. And look at this. Here's some protein, and it's going here. It's getting further modified, and then it's going here. If it's not already in the wall of this vesicle, but it's inside the vesicle, when this vesicle fuses, it releases that material. Right? So if it's a protein that's in the plasma membrane, it actually has to be in the wall of the vesicle as it's being shuttled through the cell. Now, what's going to carry these vesicles? The cytoskeleton. Yeah.
I was going to ask how the vesicles know where to go.
That's a wonderful question. Every vesicle is labeled with a protein that tells it which direction it needs to move. Because you have motor proteins that walk along the cytoskeleton, but they can only walk in one direction. Right? So then you'll have other motor proteins that will walk in the opposite direction, but the specific motor protein only walks in one direction. You're like, what does it do when it gets to the end? It has to be chemically modified to walk the other way, or just goodbye. And so they're labeled to tell which direction it needs to move. There's a label on the vesicle so that it binds to the right motor protein walking in the right direction. Yeah.
I don't know if you know what modifies the motor proteins. They're just like walking around.
Yeah, that's a good question. Yeah, I don't know. Yeah. I mean, it's probably something as simple as changing the chemistry just a little bit. So instead of being attracted to what we would call the positive pole of the cytoskeleton element, it's attracted to the negative pole. Just, cruise along. Every step that motor protein takes, by the way, requires 1 ATP, the energy currency of the cell. And sometimes this will have to take millions of steps to get from the Golgi apparatus to the plasma membrane. And you're like, that's millions of ATP. This is why every cell in your body makes and uses, on average, 100 pounds of ATP a day. Every cell in your body. I guess amazing. Yeah.
So, say for like the Golgi complex,
Uh-huh.
when it like pinches off of itself to make the vesicle,
Yes.
how is it that like the Golgi apparatus does not get smaller?
Does it? Because it's got vesicles coming from the endoplasmic reticulum that are always replacing what it's losing.
Okay.
And then the real question is, well, why doesn't the endoplasmic reticulum get smaller? And that's because what isn't shown here is the fact that the smooth endoplasmic reticulum is constantly making phospholipids. And the smooth endoplasmic reticulum is physically attached to the rough endoplasmic reticulum, restoring all of those phospholipids lost as these vesicles leave. Yeah, Davis.
Is there any DNA outside the nucleus?
Yes, but not native DNA. So it's not DNA native to this cell. But if your cell phagocytizes a bacterium, then the DNA of that bacteria ends up inside of there. But you'll typically metabolize it, break it down, and use it for energy. Yeah. All right. Well, we're out of time. It's 12:40 on Friday. So not only is our class done, but your experience with me this week is done. Unless, of course, you haven't done the quiz yet. Then your experience with me is not done. And you have until the end of today to finish the quiz.